at how does variation emerge and how could that be transmitted?
Let's return to our brown bears stranded in the Arctic to consider the impact of genetics on our understanding of evolution. Each bear is made up of cells, and if we take a brown bear cell and tunnel into its nucleus, we find DNA, the molecule with the genetic instructions for building, in this case, a brown bear written in a four-letter code. Now, the thing about DNA, it's not perfect. When it's copied, mistakes get made. Mutations, in other words, that sometimes affect an organism's traits and that sometimes can be passed from parent to offspring. So the variation at the heart of evolution, it's genetic variation. Slight differences in DNA that, for example, could make some bears a bit lighter in color, a bit more insulated against the cold, and a bit more capable of digesting fattier foods like seals.
Evolution is essentially any change in the genetic composition of a population.
Mutations are random, so they're not always helpful. But the bears with mutations that gave them some
Quick little side note, I would have loved to have like been in maybe it was a meeting, maybe it was the email, who knows. But like I imagine like whoever made this video like created it and they were like showing their boss and their boss was like, whoa. You gotta put a disclaimer in there, man. Like somebody's gonna think this is a real bear. Please. Let's just add, please note, not real. I love that. I saw that. I was like, was that necessary? But like somebody's like, no. These kids are going to see that. They're going to think there's a zebra bear out there, right? Not real.
advantage for Arctic living survived and reproduced more often than bears without them. They passed the genes responsible for those adaptations onto their cubs. Over generations, more bears inherited and elaborated on these and other changes in the DNA. The eventual result, a polar bear. And when we tunnel into its cells, we find polar bear DNA. These days, most scientists use DNA sequences to build phylogenetic trees of living organisms. Their analyses get complicated fast because DNA can mutate rapidly and unpredictably throughout the genome. So we've simplified things for our puzzles. Use the magnifying glass to compare the actual DNA of different species. We've selected small fragments from much longer sequences for you to analyze. The numbers beneath the DNA sequences correspond to the position of each nucleotide. Instead of using physical traits as we did in previous puzzles, we're using nucleotides and their corresponding position numbers. Often, one of the species will be what's called an outgroup. You can think of it as the reference to compare all the other species against. So it goes to one side of the tree. And here's the basic rule.
The more closely related two species are, their DNA is also going to be very similar.
And the more time that's passed from when two species split off from one another, the more time there's been for mutations to accumulate. So the more different the DNA will be. Think of DNA as the raw material that across billions of years, evolution has molded and built into countless forms of life. We are all related, tied to one another by the most spectacular of double helical threads.
All right, I love that video. Okay, so let's do this one together. I just want to do two. Okay, let's do this one. And this took me a second here. These took me a minute because like I didn't quite get it. So you don't have to start over. I mean, you can go right. I think you really can just go right to mission 3 here without doing that. Okay, so this tells you a little bit, like if you clicked on these things, it'll tell you about the cichlid, the coelacanth, and the frog. And that's definitely interesting. Okay, but then this is the important part. These are the DNA pieces. The outgroup is a major hint. So instead of looking at, and you can move this around, I didn't realize that at first, but remember when we were looking at dinosaurs and then they also had the random chicken and the ostrich in there. Like, what does the chicken have to do with? The question they said was, what was the chicken? What did the chicken have to do with like dinosaurs? And the common thing was they all had a wishbone, right? Here we're not looking at the outward characteristics. We're looking at the DNA. Okay, remember our four main nitrogenous bases. We had adenine, thymine, cytosine, guanine. A goes with T, C goes with G. There's no U because we're not talking about RNA, right? We're not trying to make proteins here. We're just trying to look at the DNA. Okay, so we put all these together, okay, just in the middle. Okay, so, and if you've done this already, great. Okay, and that could be wrong, right? Oh, so yeah, it looks wrong. But one thing that we have, we know that the out group tells us that that's the thing that's in common. Okay, so this cyclid here, okay, is where we've got that out group. That number one, we've got the number one part position. There's nothing there. We've got position 2. We've got position 3. Okay, so they all have C together. So we can put in position 2, they all have C. So I can put this in the bottom. Okay, and voila, there we go. Okay, but it looks like the cyclid is different. Okay, in position 3, there's the C and then there's the 2 T's. So the coelacanth and the frog go together. So I got to take this one over here. Okay, and now I put this over here like this, and voila, hopefully, boom, we've got it. So the DNA sequence of the West Indian Ocean coelacanth is closely related to the frog, and boom. So then it takes me to my next one. I would like to do just the 1 fish, 2 fish, and yeah. And so here's the thing that gave me the fits. Okay, when I first did this, because I'm slow, remember? Okay, this is position 1. Here's 2. Here's 3. Here's 4. We're not doing 5, 6, 7, 8, 9. Here's 10. Okay, so we're looking for something that they all have in common, and it looks like it's this position 3, right? They all have C's. So that C position is what they all have in common. It's much like the wishbone, right? The wishbone of the chicken and the ostrich and all of the T-Rex and all those great guys, Archaeopteryx and all those. Okay, so take a minute, do this one. I'll do it too if you haven't done it yet already. Okay.
Let's go.
It's also like, it doesn't let you do it unless you do it.
Doesn't let you do it for what?
It's locked unless you've done all of this. You can't get to.
You can't get to this one?
You go 1 and 2.
Really?
Yeah, like it's like that unlocked.
What? A lot of work. I don't know.
Well, yeah, because you did the other one.
We already did. Oh. Imagine not doing the other ones.
Wasn't here.
E-learning?
Hey, at some point, can you try to do these though? Like, cause it doesn't take that long. Like, and it's kind of fun. Like, I think it's fun.
Yeah, I like
Hey,
it.
Samuel, did you get all of them? Did you get it done? You're done, done?
Sweaty. You know what? We sacrificed
Yes,
a lot. So now, you sacrifice your life a lot to me. So everything away. What do you think it
They were
is?
like, where is your?
Second one, and the second one out.
I told him, I was like, I think I just stuck down here.
See, right, this is how you put those. So there's a 1. There's a 3. There's a 3.
Let me see.
It died.
Oh, well, you grab my chart.
Okay. I won't stop having hiccups either. So just unplug it. So then you should really get a microwave. Yeah.
If I could get rid of one thing in my house,
It would be.
it would be the microwave.
Yeah, I thought you were gonna say white. I thought you were gonna say beige.
I love the microwave. It helped me eat my breakfast this morning.
Let's go. I already got it done. Weird. I like the toaster.
Shitting is about.
Bro, Whatever happened? Do they still make toaster strudels?
Yeah.
I can never find those.
Well, do you like the strawberry?
No, cinnamon.
Ew.
No.
You are missing
Bremer.
out.
Like the
Remember
cinnamon
those
straws?
straws that have like
No, I hate cinnamon.
straws with like
Cinnamon,
little
like
then
cinnamon
you
rolls
put
and
it in
stuff.
chocolate milk, it turns to chocolate or whatever. Do they still make those?
Flavor of
I've not
Pop-Tarts.
been able to find.
I don't know the
Brown
answer.
sugar. Ew, brown
Nah,
sugar.
I don't like the brown sugar.
You buy them. I like the
Hear me out.
Christmas one, the cookie. You know what I'm saying? Like limited edition.
Hear me out. Hear me out. Cookie dough.
For what?
No.
Cookie dough tastes awesome.
Cookie dough pop tarts.
I've never had
Oh,
the
yeah.
cookies and cream.
Bro, I love those.
Those are good. The strawberry.
Oh, they busted.
What's your favorite?
I know it was a joke.
What? Oh, I heard that.
Okay.
Was it Boston?
What?
I'm almost out of level 2.
You gross. I'm not running
What?
like a
No,
gym.
like it's why can't
You
I
gross.
shop
What
anymore?
anti-venom
Fuck.
Yes, sir.
Lucas.
Didn't say that. Shut up. Negative S word. Look at this. You good?
No, my hiccups will not go away.
Yeah, how's it going?
Let's go. Hey, what's the word?
Mrs. Robinson. Yeah, I do tell
David, that's all. That's always on the thing.
It's on the menu.
Oh, big bro.
Who names their kid Garfunkel? I'd probably
Garfunkel is a great name.
probably go
Shut up.
with,
It's not.
It is too, like Oliver.
it's kind of like
Oliver and Garfunkel.
that.
Boom, perfect name for a man.
Darn it, this is too
Bruh.
hard.
So anyone named their kid. Hey, I already got
Wait
it.
till they got the name.
Taking like 10 before a month sale. I'm gonna sacrifice my life.
Sorry, I have to get a quick shop right
I will
now.
sacrifice. You might get a Popeye now.
Popeye my mask, probably.
Okay. Wait, what are sponsorship flavors? Oh, there's golden mean girl. This is so weird. There's glazed donut, Riley.
Here we go.
That's.
Pop Tart tier list maker.
I saw this one, Pop Tart was like cigarette ashes.
All right, I definitely want everybody to at least get to this one if you have.
Oh, already done.
Okay.
Riley, there's some more one.
Nah, that's
I wanted
gonna.
to talk about
That would
the
be.
DNA.
Twix
Hey.
flavored Pop Tarts.
Twix?
I
Yes,
like
did.
Twix, so Pop Tart.
Look at that tree, it's beautiful.
Do you not like Twix?
I like Trix. Wait,
Like just regular Twix?
Yeah.
Like the candy one.
Paso?
where can I buy it?
Walmart.
Walmart,
It is.
Kroger's,
Meijer.
Walmart,
Meijer.
Dollar General.
I bet you could get it at the
Target, Target,
CVS.
maybe. Dude, this one too close to strudel.
Hey, Chandler, you're on mission 5. What's the answer?
What is-
Which anti-venom will save Tyler? D.
D.
D?
D.
I
said B and I got it wrong.
It's D.
No. Yeah, Sammy. My mom does nutrition.
Oh!
I don't want to hear it. You spelled it right.
Yeah, I did, and I'm proud.
All right, folks, here's what we got. And here's the fun thing about this for me is I learned, this is something that like, I actually learned something by doing this.
Good job.
Not just like how to build a tree, but also, hey, by the way, and this is not just like, sometimes like when they do stuff, you're like, this isn't real. This isn't like real data. It's just like, they're just doing it for like an educational thing. You know, this is real data. Okay. All right, everybody, shh, just give me a second. Okay, so what we've got is a commonality. Okay, they all have the letter C, that's cytosine, in position 5 of a long DNA chain, right? Their long DNA chain is what we're looking at, okay? And then the shark is that's its only characteristic. So all of them are related at that point, okay? And this is real data, okay? This is the coolest thing. In 2013, almost 20 years ago, scientists found that coelacanths are not the closest relative of four-footed amphibians. Which species is, and that's the frog. Okay, so we thought at first that other things were related, but it is in fact the frog, and that's all thanks to DNA. Okay, so let's go back to something from last semester. Those of you who don't know, this bird seems like it's the dumbest bird on the planet. It's called the dark-eyed junco. The reason why I like it is, well, I'll tell you a couple different reasons. Here's what it looks like. It has a couple different coloration variations. And when I was in California last summer, I saw for the first time this Oregon version of the dark-eyed junco, and I about pooped my pants.
Whoa.
Okay, this is the one that we see around here. And by the way, I think they're all gone. I saw one a week ago. It was singing. You hardly ever hear them singing. And I was blocked. Yes. Let's talk about balls.
Let's talk about balls.
All right. So those of you who are not in my class last semester, okay, so all of you folks.
Testicle.
It is. It's not. Okay, so listen. So I became fascinated by this bird, not because I was interested in the bird itself, but because I was at a conference in Indianapolis several years ago, and there was a guy there who was wearing a Patagonia shirt, and this was before Patagonia kind of got like, out there. And so this guy and I started talking about his research, and he's like, "Oh, I just made a movie." He goes, "I'm an IU student, and I just made this movie about juncos." He's like, "Do you think you'd be interested?" I was like, "Heck yeah, man! I'm into birds and I teach biology." Well, it ended up that he used some of my quotes for a press release. It was really bizarre. A friend of mine in Fort Wayne was like, "I just got the Fort Wayne Business Journal and I'm reading your name in it." I was like, "What? For what reason?" And they're like, "Oh, you're quoted for this movie about juncos." I was like, "Ah, cool." So I think it's on the website. So I'll have to look at it. Yeah. So this guy made this movie, and we're gonna watch this clip again, not because I love the clip so much, but because some of you didn't see it. Okay. So we did this first semester, but there's a reason why I'm showing it to you again now, and it's not just because I want you to see it again. All right. What happened to my link?
The bird can't fly anymore because it's
Did you see it?
You guys are literally like.
Oh yeah, that'd be cool, but that's not. That's not how it works.
Like dragons.
Okay, that was.
Right.
All right, let's watch this and let's talk about it. Hey folks, if you were not my class last semester, especially if you're not my class, please pay attention.
Oh, I saw this.
Yeah.
Big balls.
Yeah.
Oh,
Thank you, Samuel.
Fat, juicy nutsack.
All right.
William Rowan was an eccentric and talented man who had many interests, including art, writing, nature, and science. But perhaps his greatest passion was birds. Originally from Switzerland, he immigrated to Canada in 1919 to pursue a career as a biologist. For Rowan, the Canadian wilderness presented a highly underexplored outdoor laboratory, filled with biological discoveries waiting to be made. He was especially curious about bird migration, the annual journeys each spring and fall between breeding and wintering habitats. What was it that triggered birds to fly such great distances every year? How did they know when it was time to go and when it was time to return? Leading theories of the time suggested that temperature and barometric pressure were the key environmental cues that birds used to calibrate the seasonal changes in their biology. Rowan saw things differently. In 1924, Rowan accepted an offer from the University of Alberta to establish a new zoology program. It was there that he hypothesized that changing day length, not temperature or pressure, was the key environmental factor that triggered bird migration and reproduction. Further, he reasoned that if changing day length in any way affected the migratory impulse, it must do so through the seasonal growing and shrinking of the gonads. Rowan is not the first biologist to consider the importance of photoperiod, but he would be the first to directly test its role in animals. He devised an experiment involving a small population of captive birds that would be housed in 2 outdoor aviaries during the winter. The control group would be kept unmanipulated to observe and use for comparison with the experimental group. The 2 aviaries would be identical. However, the experimental cage would also have electric lights that would be left on past dark into the winter nights to simulate the increasing length of days birds would experience in spring. This pioneering experiment would make Rowan famous, but it didn't come easy. University of Alberta President Henry Marshall Torrey had very different ideas about science. For Torrey, science belonged in the lab, and only the lab. In the 1920s, zoology was focused on lab studies of invertebrates, fish, and amphibians. Many regarded studying birds, especially in the field, as a foolish scientific pursuit.
The bird. It's a waste of time.
Torrey repeatedly discouraged Rowan and gave stern warnings to give up on his ornithological research. Fortunately for science, Rowan didn't listen, and he carried out his fieldwork in secrecy. As the nights got longer and the temperatures dropped, he made his day-length experiment a reality, working in secrecy on his own property on nights and weekends. The bird he chose to work with was the dark-eyed junco, a common wintering songbird in Alberta that migrated northwards each spring. Juncos were an easy choice as they were abundant in small flocks, easy to capture, and thrived in captivity. The experimental juncos received electric light well into the night for 3 months, with the duration of light increasing 5 minutes per day to simulate spring and early summer. Except for the artificial lights, the birds lived in winter conditions. In fact, it was an especially cold winter, even by Canadian standards. Every 2 weeks, Rowan recorded behavioral observations and measured the size of individual birds' gonads. And just as Rowan had hypothesized, the increasing light caused the experimental junco's gonads to increase in size. And by the end of the study, they courted and sang as if it were spring.
All right, so this is a throwback,
How much bigger than the good?
not like as big as you think. All right, so here's where we're at. We got 2 cages, right? We've got one over here with, that's the control, right? No, just natural light. Okay, we just put the birds in there. Same birds in this cage. Okay, but what's the independent variable in here? What are we changing? The light, right? We're increasing the amount of light. And then what are we going to measure? 2 things we're going to measure. Gonads, like how big the balls are, right? And yeah, when they start singing, right? When I heard the juncos singing the other day, like, and honestly,
You're gonna have to get bigger.
Whoa.
I was like,
Whoa.
no, but look, I'm gonna tell you honestly, I've never, this is embarrassing, but like I've never heard juncos singing like that. And my first thought was, I was like, oh my gosh, you guys, I was like, I can't wait to talk about this with my students because I knew that bird's gonads had gotten bigger. Now, why on earth? Now, my kids from last semester don't answer.
Sure.
Okay, Sammy, Keaton, all you guys, those of you who were. Okay, Alex, Annie. Okay, so why would you think that the testes, the gonads, were getting bigger? What's that? They're swagging, yeah. Okay. No, yeah. Why would they get bigger?
They got excited.
They did get excited
Whoa.
because why?
Knew it.
No.
They saw
Come on,
it.
not my people. Why would those gonads?
Oh,
get bigger?
summer, spring.
Smart.
Yeah, why? So, but why? What would the purpose be? Yeah, reproduction, right? They're gonna get bigger because they're getting filled with the sperm that they need to make. Now, what's the purpose of the singing? Yeah, to attract the female, right? They're not singing, the juncos aren't singing in the winter because they're not trying to get the girl. Okay, birds sing. If you heard, those chipping sparrows that we heard yesterday were males. Those were females. Those chippies are literally singing nonstop to get the girl.
Yeah.
Okay? That's what's happening. Yeah.
They poured
Okay.
their heart out in the song.
Now listen, they've done studies where like in the wintertime, they've injected birds with testosterone to the point where they sing so much that they pass out. Okay? The testosterone, they can't take it. Okay? Now listen, we've also done experiments, okay, where they've looked at the quality of song, excuse me. So birds like that chipping sparrow out there, it's going to sing for a long time. It's going to sing loud, but females can tell if he sings well or if he doesn't sing well. Okay. So because ultimately, okay, let's say this bird A sings really well and this bird B does not sing very well at all. Why would the female want the bird A that sings really well?
So then
Their children will have a greater advantage, right? They are superior. Okay, now does that mean that the bad singing bird does not get a lady? No, but they get the female settle. They're kind of like, okay, I take this guy. He doesn't sing very well. Okay, so we'll talk about the DNA and then we'll talk tomorrow about how we pass that on.